Yeah, I think we can get started now. Hi, everyone. Thanks so much for being here. My name's Kristen, and I'm a member of the AOS Student Affairs webinar subcommittee. And today our event will be about avian chemosensation. So we're really excited you all decided to join us today. We have three great speakers for you. And I'll introduce the moderator for the event right now. His name is Robert Driver, and he is currently at Duke University studying bird smell.
Yeah, thanks a lot, Kristen. Thanks a lot to everybody who helped organize this event. So it's my pleasure to host the Avian Chemosensation seminar, or in simpler terms, the bird taste seminar, bird smell taste seminar. I want to thank Kristen and the AOS Student Affairs Committee for hosting our three speakers, which have incredible research that you'll hear about, and all the participants here attending and viewing on recording later. So we'll have three talks, about 15 to 18 minutes. Two are on bird smell and one is on bird taste. And after each talk, the attendants will have the opportunity to ask questions in the chat box. And in the interest of time, we'll move from one talk, directly from one talk to the next without a verbal Q&A, but you can ask in the chat box directly after a talk. And then at the end, if there's time, we'll have a verbal Q&A for all speakers at the end of the hour. I also wanted to mention that bird chemosensation is a topic of great interest and mystery. Unlike vision and hearing, which have always been known to be critical sensory modalities in life of birds, smell and taste have been largely overlooked in ornithological history, right? So for example, we couldn't have hosted this seminar 60 years ago. And I'm saying that not because there were no virtual seminars back then, and not because there was no Zoom back then. I mean, I am saying it for those reasons, but also because there were so many biases against smell and taste in birds that there wasn't enough research in ornithology to fill up a whole hour-long seminar about bird chemosensation. So it's great progress in ornithology that we're able to all be in attendance here today and to hear this great research. So we'll move on to the speakers. And so our first speaker will be Leanne Grieves, one of today's leaders in bird smell, particularly at the level of individual or intra-species communication or recognition. And Leanne got her BS from University of Winnipeg, working on red-winged blackbirds, her MS from McMaster University, working on anis. And then she received her Ph.D. from Western University, where she worked on olfaction and its impacts on within-species communication, particularly in song sparrows. As a postdoc at McMaster University, where she also did her master's, Leanne has acquired all sorts of prestigious external funding sources for a postdoc: the McCall-McBain Fellowship, the NSERC Fellowship, one of the most prestigious in Canada, where she worked on, and their postdoc is on olfactory communication in anis. And as of last month, Leanne started, last month or last week, maybe yesterday or something. As of last month, Leanne started a Rose Postdoctoral Fellowship at Cornell. And her talk today is titled Chemical Communication in Songbirds.
Sorry about that. It looked like it was at the beginning of the slideshow on my end. So thanks so much, Robert, for inviting me and for the introduction. Thanks to the AOS and the Student Affairs Committee for hosting this webinar. I am very excited to be here to talk to you today. I'm going to focus primarily on some of my PhD research because that focuses the most directly on the communication piece. Before I dig in, I just wanted to acknowledge I am from Canada, I was born there, and tomorrow, but there's activities today. It's our National Day for Truth and Reconciliation to try to start facing and reckoning with a lot of ongoing and historic injustices that were enacted against Indigenous folks. And so I just want to say that, yeah, as of 3 weeks ago, I am now living in Ithaca, New York, and this is the traditional and I think contemporary land of the Cayuga Nation. So I'm very grateful and privileged to be here and speaking to you from here today. Oh, no. Okay. So chemical communication is the earliest evolved form of communication. And so related to that, all living organisms today use chemicals to communicate in some way, and at least to some extent, including birds. So as Robert's already mentioned, olfaction and taste as well in birds has traditionally been understudied. And this is changing quite rapidly now, so it is really an exciting time. But we know for sure, and I think all of us will convince you of this today, that all bird species that have been studied have a fully functional olfactory system. And I hope to convince you today that even bird species that we don't think about as having a particularly sophisticated sense of smell, namely passerines or songbirds, can use their sense of smell in really nuanced and sophisticated ways. And here I'm just highlighting some of the sort of poster birds for avian olfaction, like seabirds that forage out over the open ocean, finding food in the absence of other cues, and carrion eaters like vultures who can do the same, smelling their food from very great heights, and even nocturnally foraging species like the brown kiwi, which incidentally has its nares or nostrils located at the tip of the bill rather than at the base of the bill, as in most bird species, because they forage nocturnally and they actually probe through the leaf litter, smelling for food in the absence of light. But what I'm particularly interested in is how birds are using their sense of smell in these social and reproductive contexts. And for this work, myself and others have been using preen oil as a proxy for avian body odor. I want to note that this is not the only odor source, but it is a good proxy, and it's also quite easy to work with and collect. Probably most people here are fairly familiar with birds, given that this is an AOS webinar, but just to make sure we're all on the same page, this red crossbill here is accessing the uropygial gland, or the preen gland, which is located at the base of the tail. You can see an example from a Swainson's thrush of the gland enlarged here. And it's this little papilla that the oil is excreted from when birds preen or sort of probe that papilla with their bill. And just to orient you, the lobes of the gland are on either side. So that's where the oil is stored. And so for my PhD, I was really interested in learning first what kind of information was available in preen oil of my study species, which is the song sparrow. And I want to note there are a lot of other studies that have shown some of the kinds of information I'll talk about today are present. And I was able to add some other pieces of information we didn't know about, some of which I don't have time to talk about today. But for my study species, I first set out and measured, collected preen oil, and then analyzed it from a few hundred individuals from 3 different breeding populations across southern Ontario. And so I just used a capillary tube to probe the gland, mimicking the action of the bird's bill when it's preening. And just a couple milligrams of the preen oil is sufficient to do chemical analyses on this substance. So then I used gas chromatography techniques to start figuring out what is the chemical composition of this oil. And I won't get into those details today in the interest of time, but essentially you can dissolve or extract the preen oil into an organic solvent. And then when you put it in the gas chromatograph, it will vaporize the oil so that it breaks apart into its constituent compounds, which then travel through the column of the gas chromatograph at a rate that's proportional to the molecular mass of each compound. And when it exits the column, it hits a detector, and you end up with an output like this, which is called a chromatogram. And so these different peaks actually in preen oil tend to be mixtures of multiple compounds that have the same molecular mass, with larger compounds taking longer to come out and smaller compounds coming out a little faster or earlier because they travel faster. So what I'm able to do for statistical analyses is convert these complex chromatograms using multivariate statistical techniques into a format where I can collapse that information and then represent it as a single point in two-dimensional space. And so I'm going to show you some of this kind of information shortly. And so in the first case, I was really interested in species differences. And this was an opportunistic study because I had song sparrows in captivity for the behavioral experiments that I'll tell you about shortly. And I found out that somebody had brought over some female brown-headed cowbirds for an experiment that they were doing at the Advanced Facility for Avian Research at Western University. And so I just thought, hey, can I collect some preen oil? I want to see if there are species differences between the two. And we knew from prior research that there likely should be. And indeed, there are very pronounced species differences, at least between these two species. So I'll show a few figures that look like this. These are NMDS or non-metric multidimensional scaling plots. And so again, every point represents that complex chemical profile of a single individual. And in this presentation, points that are closer together indicate individuals that are more chemically similar. So you can see the brown-headed cowbirds in pink and the song sparrows in yellow are almost totally chemically different from each other. Next, I went into my wild population of song sparrows, and I collected preen oil at 2 different times of year. So in the breeding period, sort of April, May, and in the non-breeding period in the month of August. And within this wild population of birds, their preen oil chemical composition also showed a really dramatic shift between the breeding and the non-breeding period. And I was really intrigued by the fact that this seemed to be linked to sex differences between male and female song sparrows that were quite apparent during the breeding season, but that completely disappeared just a couple months later when they were finished breeding. So there's a lot of information here, and I started to wonder, whether the birds are using this information. But I want to introduce one other piece of information I'm going to tell you about today, and that has to do with the major histocompatibility complex, or MHC. So this is a really key component of the vertebrate immune system that allows, it's crucial in allowing individuals to defend against a wide variety of pathogens. And we know from the literature that most vertebrate taxa that have been studied engage in this major histocompatibility complex, or MHC-based mate choice. And this is essentially because there are caveats to this, but generally speaking, the more diverse individuals are at MHC, the more heterozygous they are, the better able they are to defend against a wide variety of pathogens, meaning they should be healthier, perhaps higher quality mates. And if you mate with an individual who's different from yourself at MHC, you're more likely to produce those heterozygous offspring that probably have a higher chance of survival and better fitness. So we know that fish and some mammal species that have been studied seem to use odor cues to evaluate MHC genotype. And we know that wild birds have MHC-based mate choice, but we hadn't really looked at the mechanism that they're doing to use this, that they're using to do this. And so we thought that it was probably odor as well. And a PhD student who's now a professor at Fresno State University, Dr. Joel Slade, had done this really neat study where he looked at preen oil chemical composition in a wild population of song sparrows and their MHC genotype. And he found that birds that were more chemically similar in their preen oil were also more similar in their MHC genotype. And I was able to replicate this finding in my captive study as well. So, kind of bringing all of this together, I wanted to do these behavioral experiments to find out whether they're actually using this wealth of information that's available. And so I brought these birds into captivity, and then I ran a series of 2-choice Y-maze experiments to test 3 main predictions in this case. So in the first instance, something I haven't mentioned yet for anyone who doesn't know is that brown-headed cowbirds are brood parasites. So females lay eggs in the nests of host species. The only parental care that they provide is in that host nest site selection. So they dump their eggs in the nests of other species, and then the other species basically get suckered into rearing the young. There are more than 100 host species that brown-headed cowbirds parasitize, but they quite commonly choose song sparrows. So we reasoned that cowbird odor should be a pretty relevant cue for song sparrows that they might respond to in some way. I did not have a directional prediction in this case because I didn't really know what they would do. I just thought they should do something if they were presented with cowbird odor. Next, I predicted that these song sparrows should prefer or spend more time with the odor of an opposite sex conspecific. These were all birds that were brought into breeding conditions, so these kinds of pieces of information relevant to reproduction should have been salient to them at this time. And then coming back to this MHC piece, consistent with the literature and what we know from mate choice invertebrates, MHC-based mate choice invertebrates more broadly, and birds in particular, I predicted that the song sparrows should prefer the odor of an MHC dissimilar possible mate, as well as an MHC diverse possible mate. And so this is, hopefully the video plays okay. This is an example of the Y-maze trial. Birds were in the start chamber for 5 minutes. Then they had 5 minutes to explore the maze, which this bird's just done. So it entered both maze arms. And then in the final 10 minutes of the trial, I would just measure how much time did they spend in each maze arm, the left and the right. And here the preen oil stimulus is placed on these cotton balls, the little white circles you can see at the end of the maze arms. So in the first case, it's not really a very good song sparrow cartoon, but if you use your imagination, perhaps they're going to avoid cowbird odor in favor of spending time with the other stimulus source, which was just a solvent. So basically a non-odorous control, or maybe they'll do something different. And so in fact, I saw a sex-specific response in which males here on the left panel spent significantly more time with cowbird odor compared to solvent, and females did the opposite. They spent less time with the cowbird odor. And I have a few figures that look like this, so I'll just orient you. The y-axis shows the amount of time birds spent in the maze arm, up to 600 seconds or 10 minutes. The larger black and white circles are the mean responses of each data set, in this case, males and females. And then the small gray circles show the individual responses. So you can see it's a behavioral study. There's quite a lot of individual variation. And then for the second experiment, I predicted that birds should prefer opposite sex conspecific odor. So I tested both males and females, predicting that males should spend more time with female odor, indicating a preference for that, and females should spend more time with male odor, indicating a preference for that. And again, that is what I found. So in this case, males spent significantly more time with the female odor, the white circle. And it's a little harder to see here because the sample size for females, unfortunately, was rather small, but females did also spend more time with male odor, the black circle. And then finally, I designed this experiment by genotyping all of my captive birds at MHC. And then for every focal bird, I would figure out an opposite sex individual that was either more or less similar to them at MHC. And I created these preen oil pairings then, where I predicted that males should spend more time with the female preen oil from an individual that was less similar to him at MHC, indicating a preference for that odor, and females should do the same, spending more time with preen oil from an MHC dissimilar male song sparrow. And then after I ran the experiment, I did this post hoc analysis where I recategorized the data according to the number of alleles each individual had at MHC. And then I predicted that the males should essentially spend more time with the preen oil from females that had more allelic diversity at MHC, and females should do the same, spending more time with males, male preen oil that was from an individual who had more alleles at MHC. So they should prefer dissimilarity and diversity. And so this figure, it's the same kind of data. In this case, sexes are pooled because they responded the same way. The first panel is showing the experimental results that were sort of the ad hoc experiment that was really designed to test. And panel B shows that post hoc analysis where I rescored the data based on diversity. And so again, here, both sexes in panel A spent significantly more time with prenoil from opposite sex conspecifics that were dissimilar to themselves at MHC. And this was similar to how they responded when I categorized the data by allelic diversity. They spend more time with prenoil from MHC diverse potential mates. So the takeaways from this, first of all, is that prenoil contains a lot of information. So this is an infochemical, but it's also a semiochemical because individuals are responding to this information, including these relatively asocial species like song sparrows. And so the song sparrows avoided or seemed to have differential sex responses to the parasite odor. And something I think would be really interesting to follow up on is to test sort of what the function of these differential responses is. So in song sparrows, males are the primary territory holder and defender. And so it could be that they're attracted to the female cowbird odor as an aggressive response. We know that song sparrows will actively try to exclude or chase cowbirds out of their territories when they detect them. So it could be that males are motivated to find and, ideally destroy these cowbirds. Being flippant, they wouldn't destroy them. They're quite small compared to a cowbird. And then females might have the opposite response. They might be, again, these are breeding condition individuals, and smelling a cowbird might indicate to a female that this is not a great place to breed and she doesn't want to be there. But at this time, that's completely speculative. Both sexes spent more time with opposite sex odor, presumably because they are motivated to find a mate to breed with for the season. And again, both sexes spent more time with MHC diverse potential mates and MHC dissimilar potential mates. And so again, they may be seeking to find a compatible mate where they can produce healthy heterozygous offspring, or a high quality mate that might provide better parental care. Because again, in this species, both sexes will help feed and rear the young. And they do both invest in parental care. So this seems to be some evidence for potentially mutual mate choice as well. So I'll leave it there. I want to thank a lot of my mentors and collaborators and trainees that helped, not just with the projects I presented today, but with a lot of other ongoing work that I have. Funding sources, field sites, and my study species. And again, I'll thank everyone for the invitation and all of you for being here today.
All right, thanks a lot, Leanne, for that great talk. I hope everybody's convinced of the importance of bird smell in these various behaviors, including mate choice, cowbirds. That was great. Like I said, you can type in the chat or hold your question if there's time at the end. And Leanne can respond if you type in the chat. But we'll move on to our second speaker, Julia Cramer, who is a leading researcher on bird taste. So Julia got her BS and her MS from the University of Constance. And her MS, this is very interesting. Her MS focused on how a pigeon's personality affects the likelihood of it getting attacked by a raptor, if I describe that correctly, which is really cool. But then Julia moved to Max Planck Institute for Ornithology in Wiesen, if I said that, I hope I said that, pronounced that correctly, to work with Maud Baldwin. And her PhD focused on taste in woodpeckers, particularly in wrynecks, which she's going to talk about today. And her PhD work on the wrynecks was published in Current Biology, and I highly recommend checking out this amazing paper on wryneck taste in Current Biology. And Julia is now a postdoc in Maud Baldwin's lab at Max Planck Institute, working on bird taste. And her talk today is titled "A Novel Mechanism Underlying Selective Loss of Sugar Sensing in Wrynecks." And I'll let her take it away. And you're muted now. The screen looked good, but the audio was off.
Okay, now it should work. I
Yeah,
hope.
that's great. Yep, you're
Yes?
good. Yep,
Awesome.
all good.
All right. And you still see the screen?
Screen's good too.
Perfect.
Mhm.
Well, thank you so much for this nice introduction. And also thanks a lot for the invitation to this exciting seminar. As Robert already mentioned, I'm gonna talk about taste today. And I'm going to talk about a novel mechanism underlying selective loss of sugar sensing in wrynecks. And wrynecks are those a bit unusual but very cool woodpeckers that you see on those pictures here. And I'm going to tell you more about them in a minute. But first, I want to give you a short background on the taste system and specifically about T1R taste receptors that we studied here. So ourselves and also most animals are constantly surrounded by potential food sources in the environment. And we have to make choices on what to consume or what to eat because it might be beneficial and give us energy and might be nutritional. But also we want to avoid toxic or harmful compounds in our food. So we have to choose wisely what to eat and what not to consume. And the taste sense is a very crucial sense for us to make those decisions. And there are 5 main basic taste modalities that you probably all know. So there is sweet and umami, so the taste for sugars and the taste for amino acids that are usually appetitive. And then salty, bitter, and sour that are usually aversive. Salty depends a bit on the concentration. So how does that work? Like in mammals, on our tongues, we have those specific structures, folded structures called papillae, and they contain taste buds. Taste buds are basically bundles of taste cells. And those taste cells have little receptors sitting on top. So in the case of sweet and umami taste, these are G protein coupled receptors called GPCRs from the T1R family, so taste 1 receptors. And if we eat something, then the molecules of the food get washed over the tongue and they can interact with the receptor and then also activate the receptor, and we will perceive that specific taste. In case of sugar, for example, we will know this tastes sweet. And in case of those T1Rs, these are heterodimeric receptors, so they contain of 2 subunits. And for sweet taste in mammals, this is T1R2, T1R3, and for umami, this is T1R1, T1R3. So now you might wonder, why is sweet taste perception in birds particularly interesting? Well, as I said, like in mammals, we have those 2 different receptor pairs forming functional receptors for the 2 tastes, sweet and umami. However, in birds, the gene for T1R2 is lost, and that is for all birds. So they have lost it very early in an ancestor, probably in a dinosaur ancestor. So the question is whether and how birds can actually taste sweet, because some of them really rely on very sweet and sugary stuff, like hummingbirds that drink nectar, for example. So it was found that, interestingly, in hummingbirds and also in songbirds, their original umami receptor has natural occurring mutations that repurpose that receptor to also taste sweet. So those bird species have the T1R1, T1R3 subunits forming a receptor that is necessary to taste both tastes, umami and sweet. So birds can actually taste sweet, or some birds can. And interestingly, between songbirds and hummingbirds, it was found that this is the same receptor, but it's still different regions in the receptor that are important to have mutations that have this convergent evolution of sweet perception. So now, of course, there are many more different bird species, and one of them are woodpeckers, and they belong to the group of coracimorphs, which are early land birds, and they haven't been studied so far. So when you think about woodpeckers, you probably think about something like you see on this picture. So there's a woodpecker sitting at a tree trunk and searching for insects, which is actually true. So 90% of the diet of woodpeckers is usually insects, and here you can see a phylogeny with the more than 200 woodpecker species, and we mapped the level of insect diet on their tree, and you can see they actually feed a lot on insects. So, however, surprisingly, there are different sweet food sources that they also like. So there, for example, is the sweet tree sap that some species really enjoy. Also, nectar is something they eat. Like here, you can see an acorn woodpecker feeding from a woodpecker feeder, from a hummingbird feeder, which contains pure sugar water. So they seem to be attracted by the sweetness of it. And also, a lot of species really like to feed on fruits. So you see there's different sweet diets that woodpeckers seem to consume, and actually the consumption of sugars within this woodpecker families is quite widespread. So if we again look at the phylogeny of the woodpeckers, you can see that there are quite many species that actually have a high proportion of sweet in their diet. And just to orient you a bit on that tree of the woodpeckers, there are the core woodpeckers, the so-called Piscinae, and then the strictly insectivorous piculets and wrynecks at the base of the tree. And looking at this pattern here, it looks like there might have happened another gain of sweet tasting in the core woodpeckers, in the Piscinae. And to test this hypothesis, we picked two different species. One is the wryneck as an outgroup because it has a very specialist diet, almost exclusively feeding on ants, and it does not contain any sugars in the diet. And we compared that to the great spotted woodpecker, which is a generalist regarding their diet. So they also have a lot of fruits and also sap in their diet. And as a first step of this study, we were interested in seeing if we could see how these natural history records are reflected by reference assays on like what they actually like to feed in two choice assays here. So what we did was we caught wild birds for woodpeckers. We went to Austria and we caught 10 there, and that was pretty easy because they're quite common. However, for wrynecks, this is a bit more tricky. They are much rarer. But we were lucky enough that we could collaborate with a ringing station in Italy on an island in the Mediterranean Sea, which is a migratory stopover site. So over the course of 3 years, we were able to also catch 10 individuals of wrynecks and like perform those taste tests with them. We then kept the birds in cages for a couple of days and released them back to the wild. And they were doing great afterwards because sometimes we even recaught them. So they really seemed to come back to us. However, the experimental protocol was the same for both species. So we would remove the maintenance food for a little while just to increase feeding motivation. And then we would present a stimulus, sugar, for example, and a control food that is non-sweet, and see if they show like any preference and how much they feed from each side. And then we would later on like switch the sides for control and stimulus food to control for side bias. Just to give you an idea of how that looks, here you can see a great spotted woodpecker approaching the setup, and he seems to be interested in drinking, so he starts sampling. On the right side, there is water, and on the left side, there is sugar solution. It's crucial for our experiments that the bird tries to sample from both sides, because then we see if they actually can taste the difference or not. Now you can see that the bird switched to sugar, and it seems to really like it just by looking at it, because it won't really stop drinking. Like it will stop drinking eventually, but you can really see a strong preference already just by like looking at the bird. For wrynecks, it was a bit harder because they rarely drink. So we found in the end that they really like mealworms. And what we did with them was that we sugar-coated mealworms and we presented like those to them. So there was one sugar-coated version, one control version. And if we look into the sampling like numbers of both species, we find that actually the great spotted woodpecker statistically significantly prefers sucrose over water, whereas wrynecks do not. And importantly, if we test the same with an amino acid alanine, we find that both species show a preference for the amino acid over control. So now we wanted to take that to the molecular level and see if we can actually check if this has a molecular base, and wanted to test the original T1R1, T1R3 umami receptors in functional cell assays. And we can see that both species, the wryneck and the woodpecker, respond to amino acids. However, only the great spotted woodpecker also reacts to different sugars, but the Reinecke response is not there. So they can't detect sugars with their umami receptor. And that fits quite nicely what we saw from their behavior. And like that, we also uncovered that there is another convergent case of this repurposed umami receptor to also taste sugars in woodpeckers. But then we were interested to see if we can maybe find the key residues for that, or regions like that that play a role in this convergence. And first, we were interested to see when this actually evolved. So we did ancestral reconstructions of our receptors or receptor sequences. And we did one ancestor of all Charasimorphs, those land birds, which is here in green, the ancestor 1. And then we had 2 other ancestors, one ancestor of all woodpeckers and one ancestor of just the core woodpeckers, where the great spotted woodpecker belongs to. So now when we tested those receptors for their response to sucrose, we find that the very early Charasimorph ancestor didn't have a response to sucrose. However, and surprisingly, that both the ancestor of just the core woodpeckers, but also of all woodpeckers, had a sucrose response in their receptor. So if you remember that phylogeny that I showed you before with all the sweet diets that the woodpeckers consume, we thought that the gain happened in the core woodpeckers. However, now from those receptor assays, we see that the gain already happened in the ancestors of all woodpeckers, and there wasn't a gain in the core woodpeckers, but there was a loss in the Reinecke. And this might be due to their very specialist diet that they probably don't need to taste sweet anymore. But it's also a very rare case of sensory reversion, because all birds had lost their ability to taste sweet. And then later the woodpeckers gained it again, and then the Reinecke lost it again. So this is very rare to see. And we were excited if we could look into the actual key regions of that. So if you look at the receptor structure, they contain 3 specific elements, which is the venous flytrap domain that usually interacts with sugars and ligands, then the cysteine-rich domain and transmembrane domain. So now if you look into the wild-type receptors of the woodpecker in yellow and the Reinecke in blue, and we test those, we see that the woodpecker responds to sucrose and the Reinecke receptor does not respond to sucrose. Now, if you look into mixed pair receptors of having just the Reinecke T101 and woodpecker T103, we see that this combination is enough to have the sugar response. And even more interesting, only the transmembrane domain of the woodpecker T103 seems to be enough to have this sugar response. And importantly, the reverse chimera of a woodpecker with a Reinecke transmembrane domain of T103 does not have this. So we see here that the transmembrane domain only is responsible for this sugar response and for the loss in the Reinecke, which is very different from songbirds and hummingbirds, where the residues were scattered all over the receptor, basically. So now looking into this specific domain that we just found, we decided to create receptor chimeras with a Reinecke background and having quarter chimeras of this transmembrane domain with woodpecker sequence. And you can see here that we, with only the last quarter of the woodpecker is sufficient to recover that sucrose response. And if we cut that into 2 halves, then the first half of this last quarter is enough to have the sugar response. So it's a very tiny chimera, actually, in a very tiny region. So we call this chimera TM6 chimera because it's in the transmembrane domain 6. And between the 2 species, the great spotted woodpecker and the Reinecke, there are only 5 amino acids different in this region. So what we did was creating point mutants to knock out basically the woodpecker sequence here. And we find that, interestingly, only in 1 knockout is enough to abolish the sugar response. So a change at position 763 to alanine is sufficient to switch that sugar sensing in the Reinecke off. And that is pretty cool. So here you see a homology model of the receptor transmembrane domain, and the red residues here are basically acting like a micro switch in the Reinecke to switch that sugar sensing off. And it's pretty cool because this also lies in an allosteric site. that is known in other species, also in mammals that might interact with allosteric modulators, positive or negative. And you can also see that it's in this transmembrane domain 6, TM6, that is close to the T1R1 TM6. So we think that this residue might actually break up some crucial interactions to form the stabilized conformation of this receptor when it's activated. So that one point mutation in the transmembrane domain is sufficient and explains the loss of sugar sensing in rhinex. So as a take-home, we found here that woodpeckers also convergently repurpose their umami receptor like hummingbirds did. So woodpeckers can also taste sugar with their umami receptor. And rhinex subsequently lost that trait again. And that loss is due to one single residue in the transmembrane domain of T1R3. And we think that this might be closely related to their specialist or generalist diet, because in case of rhinex, I mentioned before, they really feed mostly on ants. So it might just be a retuning after they shifted their diet to exclusively like ants, that they just don't need to taste sweet anymore. However, there could be also other traits involved. For example, some bird species like starlings, they don't have the ability anymore to digest sucrose or sugars, and this is very harmful for them. So there could also be something in the digestive system of the rhinex that helps them to not taste sweet and go for it and actually protect them. But we don't know this, so this is subject of future studies. But yeah, so this was a paper published by my amazing co-authors, Elliot Miller. I want to mention also Massimiliano Cardinale and Leonida Fusani, and of course my great research group and especially my supervisor, Dr. Maud Baldwin. So she was giving me the opportunity to work on that project. And with that, I'm happy to take questions either in the chat or later on if you have any. Thank you.
All right, great. Thanks a lot, Julia. Amazing research. I'm glad we got the wry-neck head neck movement in there at the end as well in that slide. Thanks a lot for the talk. And we are working on Haoran's talk because there were some serious technical difficulties, and he has sent me a file. However, the file is too large to download in the time that we have left. So perhaps we'll post this on the AOS website after so people can view it. I just want to give a plug to Haoran anyway. Actually, I will say, because we have a little bit of extra time, we can have a verbal Q&A just after this. I'm assuming everybody's okay with that. But I'll just say Haoran is really great, and you should check out his research. And he does most of his it's in different areas of genome biology, but he's done a lot of work on olfactory receptor molecular evolution. Particularly, he published the first expression study of the bird olfactory epithelium in multiple species of birds in a single paper. It's in BMC Genomics, and it looks at night heron versus egret olfactory receptor expression, so controlling for nocturnality there, and to look at how smell is affected by a transition towards nocturnality in the night heron. He also has this paper that just came out this year looking at a very high quality great bustard genome in communications biology. And he also discovered a massive olfactory receptor tandem repeat cluster in this new chicken telomere to telomere genome. And that's what that's one of the things he was going to talk about today. But we'll figure out a way to get that research to you guys, since it is very fascinating, I think. So what we've got 10 minutes or so here. If anyone wants to ask Leanne or Julia questions, I think this would be the time to do it, either in the chat or if you want to unmute, right? Just making sure that's okay with AOS. Yeah. Okay, cool.
Yes, absolutely. And we will try hard to get Haoran's talk. Maybe we can even edit it into the same video as this recording.
Oh, that would be
before we
amazing.
share it.
Then no one will know about everything we went through.
Yeah, but thanks so much to Leanne and Julia. So yeah, please, anyone who wants to ask questions live or ask in the chat and we can answer them live. Yeah, Sophia, go ahead.
Well, I have a question for Leanne. I know that like in a lot of mammals, they respond really strongly to like if you put on, you know, like cougar urine around, like the mammals will really avoid it. If you put up like mounts with preen oil on them, will birds react similarly, do you think?
That's a good question. There have been a couple of predator studies to see if birds will avoid nest predator odor, for example, and the results have been mixed in those few studies that have been done. In terms of how birds will respond to the odor of other birds, I think there's even less research on that. Danielle Whitaker's group had sort of a preliminary pilot study that used a different brood parasite odor applied to nests, and there was some really compelling evidence that the females, the host, well, the would-be host, I guess, would abandon those nests more often than not. But in terms of other kinds of behavioral responses, I don't think that's been explored, but it's a good question. Thanks.
Oh, thank you.
I have a question for Julia, if that's okay. I might have missed the slide a little bit because I was answering a question, so I apologize if I've gotten something wrong, but
No.
I thought it was really cool when you showed that maybe that receptor loss was because of the specialist ant diet. And then I immediately thought of the flickers because they prefer to eat ants. And I thought, oh, maybe they'd have the same thing, but then they haven't had that loss. And so I was curious why you think that might be, or maybe I missed something. Or maybe they eat more other things than I realize. I don't know.
Maybe they do, but yeah. So I think it's a very cool question and it's very interesting. We can't ultimately answer it, I think, with our study. For the Rhinex, at least, we think, so the thing is that based on their mutations, we think that they probably can't distinguish anymore between sugars and amino acids. And it's just very appetitive, like both of it might taste just the same, but appetitive to them. So as we know that some of the birds we study in other projects really have issues with their digestive enzymes, and they might even die if they consume sugars. This might be the case for Rhinex as well. So it might be actually harmful for them to feed on sugars, and they can't distinguish anymore, probably. So that might be just like a mechanism they evolved to kind of rescue themselves from that. But we don't know. It's just a hypothesis, but it's one of the main things. Otherwise, it could be also something that just evolved in the course of evolution because they really don't need it anymore. And having just one mutation, switching the sugar sensing off in the Rhinex, might be something that just happened. It doesn't like require a suite of different mutations, you know. So yeah, and since they only feed on that, they might not have retained the sugar taste. But it's interesting to, of course, like look into different species and see what they can actually taste or can't taste. And potentially like other woodpecker species also have like this mechanism, but we don't know so far.
Thanks.
So interesting. Jay, go ahead. You have your hand up.
Thanks. Yeah, I also had a question for Julia. And again, also apologize if I didn't quite catch this, but what do you think is the reason why the umami receptor just keeps getting repurposed for sweetness? Is it something about the receptor, or is there some sort of like similarity in the molecule and how the molecule interacts with those two receptors that's similar?
Yeah, thanks a lot for that question. It is actually a very similar molecule because they are both G protein coupled receptors, and they are very conserved across all animals usually. So this is very interesting because compared to olfactory receptors, for example, there are so many. But for those taste receptors, we only have those. And it's always those heterodimers coming together, like for other tastes, like for salt or sour, for example, it is ion channels. But for sweet and umami, it is those actual G protein coupled receptors, and they interact with the ligand, may it be like amino acids or like sugars. And because birds don't have the gene for the second subunit, they are really unable to form that sugar receptor. So they can't just have it. But it seems like the umami receptor is a rather easy way to recover that sense, like the sweet taste abilities. But we can't, of course, rule out that some other birds might have evolved like secondary pathways or mechanisms to taste it. Like there are glucose transporters that we know of, like in other species, for example. This might also play a role. But I think, yeah, for your question, it might just be, let's say, an easy way that evolution can take to restore the sense that they lost, because you can't regain the full gene. It's not pseudogenized, but it's really like lost fully. So yeah.
Very cool. Thank you.
Guys, great questions. If I could just jump in here, Haoran's talk is now working for me. So it's only 10 slides. I think we're going to go slightly over time. So no pressure on anybody, but it would be great if I could try to play this in the last few minutes we have.
Yeah, that sounds great, Robert. And if folks have to leave, we'll be sharing the recording to everyone who registered, so you could catch up on the end if you have another commitment. But yeah, if we can just play Haoran's talk, that would be great.
Feel really sorry that I only can use this video to share my work, and happy Mid-Autumn Festival today. It's my pleasure to share my research about olfactory receptors in this seminar. I'm from Xiamen University. My topic is weighing olfactory trade-off and a tandem repeat of olfactory receptors in birds. The numbers of olfactory receptors usually positively linked to animals' olfactory ecological adaptations, such as dog had more than 800 olfactory receptors, and elephant, the most well-smell animal, have about 2,000 olfactory receptors. In human, over 16% olfactory receptor pseudogenization due to the weighing olfactory trade-off, the same as in the trichomyces primates and ferrets. Indications of smile nocturnal brown kiwi and kakapo have larger olfactory receptors than their diurnal closet relatives. Total numbers of olfactories was larger in these two nocturnal birds. Some bird species exhibited evidence of olfactory receptor genes expression, including the little egret, parrot, and the chicken. As for the little egret, the expression of olfactory receptor is unusual. In ordered birds, unordered birds, usually lambda egrets, and nocturnal ordered birds, usually lambda horns. We compared two egrets and two horns. Nocturnal horns had less olfactory receptors than diurnal egrets. This is unusual, and this is inconsistent with previous couple and kiwi research. Thus, we are wondering why diurnal expression, or we can say why nocturnal contraction. Then we compared intact and expressed olfactory receptors in olfactory tissue. We found reduced total intact and expressed olfactory receptor genes in nocturnal horns. The expression levels of olfactory receptors were low in olfactory tissues. We strongly suspected that if it is the trade-off between weighing and olfaction caused olfactory receptors contraction. Fortunately, these two horns had different visual nocturnal adaptations itself. The black cloud light horn, they rely on weighing for foraging. However, the both-billed horn not fully rely on weighing, but also rely on tactile. Thus, these two species can help us understand weighing adaptations in horns. We found weighing genes related to retina development and eye size were under positive or relaxed purifying, which can be considered as weak positive selection in black cloud horn. Whereas weighing genes were under relaxed or intensified purifying selection in the both-billed horn, such as we can see the retina development related CCDC166 gene showed positive selection in the black cloud light horn, whereas showed intensified purifying selection in the both-billed horn, suggested that the two species were not adapted to nocturnity with different genetic mechanisms. This would suggest a trade-off between olfaction and weighing. Now we return to expression of olfactory receptors in some birds. The expression mechanism are revealed for a long time until new sequencing technology applied in birds. Recent chicken complete genome showed that olfactory receptors tandem repeat in the chromosome 29, which incomplete assembly in previous several assembly variants. Personal opinion, this is a challenge for olfactory receptors research in birds because of most ecological adaptations analysis of olfactory receptors performed on NGS-based reference genome. The problem is that this chromosome missing in previous assembly due to scaffolding field. Olfactory receptors tandem repeat in the relative high repeat and the GC content region, which with high levels of methylation and the NGS and pachydiasis sequencing coverage were also known. It was vital to olfactory receptors ecological adaptation research because we usually counting olfactory receptors numbers and perform downstream RNA-seq or WGBS would be subjected to by low coverage. Okay, let's do a summary. In nighthawks, diverging adaptation of weighing-related genes and reduced and low expression levels of olfactory receptors should allow trade-off between olfaction and weighing, and the tandem repeat of olfactory receptors in hard family chromosome. I think it is challenging for counting olfactory receptors for olfactory ecological adaptation research, and the downstream regulation and epigenomics analysis is also a challenge because of the low sequencing coverage. And so thanks for listening. I would like to thank AOS and Dr. Robert called this seminar, and I would like to thank my mentors, Professor Chen Xiaoying, Professor Xu Luohao, Professor Shen Yingjia, and Dr. Zhou Xiaoping, and thanks all group members in Southwest University and Xiamen University. And I feel really sorry that I only can present my research through this video. Sorry, and thanks again.
Yeah, we have a couple minutes left. I think Ashley had their hand raised earlier, if you still have a question.
It didn't mean to interrupt the Q&A. I guess but my question was, so you've looked at you must have thought of this, Julia, a bunch of times. You looked at the piculate sequence, right?
Not at the piculates, no.
Oh, okay.
Just the
so we don't know if the piculate receptor has that mutation or not?
No, we don't know. We don't know if they have this mutation. Yeah, that's true. But we know that from ancestral reconstructions of like all woodpeckers, we know that the all woodpecker ancestor had the ability to taste sweet. So this is something we know. So it might have evolved like twice, maybe, but that we don't know. Yeah.
And I think we have time for one more question. So Siddharth, if you have your hand up, go for it.
Yeah, hi. Great talk. I have a question for Julia. Yeah, really nice talk, very interesting. I don't know too much about sensory receptors and all of that, but I do have a question. Do you think this could have happened in any other group of birds? Do you think some other group of passerines could have lost their sensory receptor, like what you have found out? Do you think that's a possibility? I mean, especially for these insectivore specialists.
Yes, so we don't know so far, but we think it's definitely a possibility, and it would be also a very, let's say, elegant way of switching off that one taste modality. Like, as I said before, we strongly think that maybe it's a mechanism that might also help birds that have issues with their digestive system. So in case this is going on, for example, in some songbird species, which like starlings, for example, they do belong to songbirds, but apparently they really like sugars and it's harmful for them. So we think it might be a mechanism that is applied in different like bird species. If it's exactly the same residue, we can't tell, of course, without like going through the receptor sequences. But yeah, we are currently also comparing like various species and looking into it, like also outside of songbirds. I'm looking into hornbills and mousebirds, for example, and there's also some quite cool stuff going on there. However, we didn't find any mutations just like switching off selectively the sugar sensing while retaining, of course, like the amino acid sensing, which is pretty cool that they just didn't lose the full functionality of their receptors, but just switch one taste modality off. But yeah, it's definitely a possibility, and we're looking into it.
Yeah, great stuff. Thanks.
All right, everybody. So it's 11:02. Actually, we have one. We're gonna fit this one question from the chat in, and then we'll say goodbye to everybody. As a high school freshman, okay, who uses English as a second language, wow. It's a little hard for me to totally understand the webinar. I will later, but I have some questions closely related to what we were talking about today. If I want to pursue a career with birds, any suggestions for my university and major choice? I'm going to study abroad. Just message you, just message back. Well, that's a loaded question, but there's some great labs here. If you're interested in chemosensation, Maud Baldwin's lab, where Julia comes from, and McMaster University. I don't know if you guys want to take this general question, but maybe we'll contact you, Shannon, if you have the contact information that you want to send. Maybe we can get to you. Actually, she sent this directly to me, I'm realizing, so you guys don't see this question, but it applies to everybody.
What I will do now, Robert, is I'm going to send a link to an AOS page that's all about resources for students.
Okay, great.
So they can check that out. And yeah, we are over time, so I just want to say thank you so much to all of our speakers, and thanks so much to Robert for moderating. And if you have any questions or ideas for future webinars, feel free to reach out to us at studentaffairs@americanornithology.org, and we will be in touch soon via email with the recording for this event, and hopefully that includes all 3 talks. And yeah, we look forward to seeing you at our next webinar. So thank you so much, everybody.
Okay, great. Thank you so much, everybody, for attending, and to the speakers. Thank you so much.
Thanks, everyone. Bye.
Thanks for attending.
Thank
Bye.
you.
Thank you.